Hey everyone, welcome to our special edition of Student-Led Pass for this week in 1035 MSc. Today we're going to be discussing sickle cell anemia and balancing selection.
Thanks, Jordan. This slide addresses the main structural components of a red blood cell and some key features, including its large surface area for increased gas exchange and small shape, allowing it to navigate through the capillaries. So what is sickle cell anemia? Sickle refers to the mutated shape of the cell. Here are some consequences of that mutation. Anemia refers to the low red blood cell count, resulting in poor oxygen saturation around the body. Here is a reference sheet which has all the key concepts related to this content, so feel free to screenshot and refer back to it at any point in time.
The HBB gene that codes for the beta globin is on chromosome 11. In this mutation, an adenine nucleotide is substituted for a thymine, which produces valine instead of glutamic acid in the beta globin chain, causing it to fold differently. Bonus question, what is this type of mutation called? That's right, it's a non-conservative missense mutation. So why does it fold differently? Well, valine and glutamic acid have different properties, and these different properties cause the amino acids to interact with their environment differently, influencing how the chain arranges itself. Misfolding of the beta globin mutates the entire hemoglobin into HbS. HbS molecules in the red blood cells polymerize in low oxygen conditions, forming rigid rod-like chains, distorting the red blood cell into a sickle shape. If only one S gene is inherited, the individual is a carrier of the disease and can also exhibit codominant expression of the two genes under certain conditions, like high altitude or dehydration. Okay, complete the following activity. Yeah, that's right. So an A and an S genotype will be a carrier, an S and an S genotype will have sickle cell disease, and an AA genotype will have neither. Well done.
Thank you, Sienna. So the reason S allele is maintained despite causing sickle cell anemia is due to a protective advantage against malaria and trait carriers. This phenomenon is known as balancing selection. Here we see regions with high malaria prevalence showing a direct correlation with higher S allele frequencies. Plasmodium falciparum, the most common and deadly malaria parasite, is transmitted by infected Anopheles mosquitoes. Once inside the body, the parasite develops in liver hepatocytes before infecting red blood cells and spreading. While the exact mechanism of sickle cell trait-mediated malaria protection remains uncertain, some studies suggest that infected AS genotype red blood cells can deform into the sickle cell shape, which may trigger early destruction in the spleen. Additionally, sickle cell trait carriers may also gain an accelerated acquisition of malaria-specific immunity. Here we see two allele frequency predictions based on fitness scores for AA, AS, and SS genotypes. Which population would you expect to have a higher clinical malaria rate? That's right. The muscle lip population has a high clinical malaria rate, which is why there's an increase in S allele frequency.
Thanks, Liam. I'll now be running you guys through some brief revision on the categories of sickle cell anemia symptoms. Once we've done that, I'll test how well you've been listening to the mini lectures with a matching game. In the table below are our main categories of sickle cell anemia symptoms. As you can see, that mutation on chromosome 11 has quite the impact, as those symptoms there don't even brush the surface of possibility. The graphic alongside the table illustrates some of the main areas affected by these symptoms in colour and less affected organs in grey. Now that we've covered that, let's test your knowledge. The game is simple. Match the symptom to its physiological cause as fast as you can. When you're finished, please read the note below and we'll go through the answers. Alright, it's time to go through the answers. Wow, you guys really must enjoy that content. 6 for 6. The end of trimester exam will be an absolute breeze then.
Thank you, Jordan. Treatments range from symptom management to curative therapies. Pain management, hydration and oxygenation, as well as infection prevention, can all improve quality of life for many patients, as well as hydroxyurea and chronic blood transfusions. Bone marrow transplants offer a cure but come with risks, limited donor availability, graft versus host disease, and potential infertility or secondary cancers. New therapies are being developed, including gene therapy, which aims to provide long-term solutions by directly correcting the genetic mutation. CRISPR-Cas9 is a promising gene editing technology that corrects the mutation causing sickle cell disease. It offers a high success rate while avoiding donor dependency, while CRISPR-Cas9 reduces long-term complications. Risks include graft versus host disease, infertility, potential secondary cancers, and unintended genetic effects. Gene editing raises concerns about accessibility, long-term safety, and ethical dilemmas surrounding human genetic modification.
All right, thank you, Noah. I appreciate you guys joining in for today's pass session, and we hope to see you guys next week.